cytotoxicity and phase contrast microscopy, we observed some apoptosis is occurring. But how the apoptosis induced by unique IS2, we've done by modulation and activation of the P53 and modulation of the Bax BCL2 ratio, release of cytochrome C and activation of the caspase 3 and cleavage of the PARP protein. And of course, HSoup is giving the positive response. Up to now, we discussed about live probiotic, but not on this protein, because here completely it's a protein. How it is responsible? What are the things are playing to give the effect of these things? To observe that one, we went for metabolomic analysis of unique IS2 treated HSoup. To do that one, we selected culture supernatant and this heat shock supernatant, and we extracted the metabolites using methanol and evaporated and analyzed for GCMS analysis, and top 200 components we blasted, and we found the following results. The first one is that one control culture supernatant, only naphthalene and anthracene degradation and purine metabolism components were observed, but whereas in HSoup, biosynthesis of unsaturated fatty acids, styrene degradation metabolites, butyrate metabolism, and pyruvate metabolism and fatty acid metabolism components we got in the HSoup fraction. So these are how this HSoup fraction is responsible. It produces short chain fatty acids like lactic acid, estetic acid and butyric acid, and unsaturated fatty acid metabolism is also observed. So they produce polyunsaturated fatty acids and conjugated fatty acids. And the last one is styrene degradation metabolism. It is useful for the energy, it produces energy for the pyruvate metabolism pathway. And last one is butyrate metabolism. It helps to maintain colon cancer cells. So of course, HSoup is responsible for these things. And next we want to do is the in vitro work, and we want to go for whether the effect is it useful in animal model or not. So we are going to do that study soon. And these are the clinical studies we've done on Bacillus coagulans unique IS2, acute diarrhea, vaginosis, lower cholesterol, and functional abdominal pain and liver cirrhosis. These are the glimpses of our factory pictures. We have highly qualified people to do work, and we, as per Alliance person, the sir, he told about the GMP of probiotics. Like that, we are also maintaining. And we have dedicated facility for all different probiotics. Like Bacillus, we have separate block. Lactobacillus and yeast, we have separate blocks to manufacture to avoid the cross contamination. These are the quality systems we have in Unique Biotech, designed to meet ICH, PICS and ANVISA guidelines, Apex quality documents in line with ICH quality manual, water manuals, we have GMP and validation master plans, and as per ICH and FDA guidelines, we have documentation with the process validation, and as per he told stability like SH guidelines, based on the stability of the product, shelf life of the product, we have different stability analysis also, and policies and procedures in line with international quality requirements, and management oversight review over critical quality management systems of the part of the Unique Biotech. Thank you.
Thank you, Dr. Suvarna. I think questions we can take later on only. Should we wrap up the session then, in that case? Okay.
No, there is one question. Mary, you want to take a question?
Thank you for your presentation. One thing I've never been clear on with the use of a spore as a probiotic is what growth stage we would expect in terms of germination in a human. And so are the spores basically transiting through the human, or is there evidence that there actually is germination in humans? And if so, to what extent, and what's the ratio of spores versus vegetative cells that are ultimately found in the feces?
Yeah, we understand the process. We've done one study in vitro model, ma'am. Intestinal model with ProDigest we've done, whether the whatever ingested spores are germinating, where they are germinating, we tried, and we gave 2 billion cells initially, and after intestinal, of course in gastric tract, there is no germination of the spores because of the adverse conditions of the nutrients and all. But whenever it went to intestinal condition, so whatever 2 billion spores, they increased and germinated, and it releases in colon. Up to colon also we studied, and we observed short chain fatty acids also. So germination is occurred.
So you have evidence that in humans you can isolate out vegetative cells.
We isolated from contaminated human soil, ma'am, feces.
From feces, so
Yeah.
not in an in vitro model, but actually from humans. So
Yeah.
you do have evidence that they germinate.
That we
And-
don't have, ma'am.
You do
We
have
isolated
evidence.
from human feces, contaminated human feces. We isolated.
So you isolated vegetative
Yeah.
cells from the human feces, but
Yes.
exactly where in vivo that occurs, is it in the distal colon likely, or where is that likely to occur?
Okay.
The germination, because you're feeding spores, correct? So where does the germination and the
Yeah,
vegetative cell?
intestinal. It's
In the where?
in the intestinal condition, ma'am.
Under intestinal conditions, but the intestine is diverse. So where in the intestine is this likely to occur? Is it in the distal colon? Is it? Do you know? I mean, I think it's relevant in terms of what type of physiological
Yeah,
benefits you
we've
would
done that
expect.
study, ma'am. It's study is going to be published with ProDigest. We've done that work.
Okay, you have done that work.
Yeah, report we have. So where it is germinating, the report.
Okay.
Just to clarify, when you say contaminated soil, so is it like the
Feces.
human volunteers were fed, and then you collected feces of those
Yes.
humans?
Yes, sir.
So it's nothing like contaminated, like regular
Yeah.
stool, and they were vegetative. They were in the vegetative condition.
Yes.
You recovered them.
Yes, sir.
Okay.
Have
then
you
in-
quantified that? I mean, do you know whether or not 10% were recovered or 0.1%? I mean, do you have an idea of, is it the majority of these spores that are then germinating, or is it a small fraction?
Actually, we gave initially 2 billion, ma'am. After vegetative cells germinated, the viable count is increased.
It increased?
Yeah.
Okay. Thank you.
Thank you, ma'am.
I think that's okay. Then maybe one or two quick comments from our coach.
Now I request Dr. R.R.
Yeah. 1 minute, just. Would you like to comment on this?
So we come to the end of this session, and we have had an amazing line of speakers, and we had very diverse topics right now. We started with the lecture on the GMP, and we ended on one of the probiotic organism which they are looking at, and Dr. Kocher was there. She gave us the link between the gut and the brain, which we have been hearing since yesterday. So since this is the last session, so we feel that we will go back home with a little bit more enriched, with more brainstorming. We can come up with new topics to work around, and we might have answers next time when we meet again. Thank you all of you for your patient hearing.
Okay, so I also thank the audience for being very patient, and we are already delayed. So maybe, I mean, the session could have been more lively because unfortunately we never had time to put questions before the audience, but I invite all the audience to please interact with the speakers during the lunchtime, and still if you have any doubts, you can pass on the questions. We'll take them up during panel discussion. Thank you so much.
I request Dr. R.R.V. Singh to present the mementos to Dr. K.P. Kocher and Savarna Lakshmi. Madam? वो तो दे दिया। Dr. Kocher.